Welcome back to the Genetics Lecture Series. So far, we've spent a lot of time learning about nuclear DNA and its association with human health and disease. In today's podcast, we are going to focus on mitochondrial DNA and the features of inheritance associated with the mitochondrial genome. The objectives for today's podcast are to review the composition of mitochondrial DNA and its relevance to the cell, to define homoplasmy and heteroplasmy, and to highlight what makes mitochondrial DNA inheritance unique. These are the new terms that you'll be introduced to in this podcast. Mitochondrial DNA is a circular chromosome that contains only 37 genes. Instead of paired chromosomes as seen in nuclear DNA, there are many copies of mitochondrial DNA in each cell that proliferate independently from each other. The number of mtDNA copies in each cell varies by cell type. For example, the oocyte at fertilization has 1 million copies of mtDNA, a sperm cell has about 100 copies, and red blood cells have none. Mitochondrial genes encode for 13 subunits of enzymes that are involved in oxidative phosphorylation, the process by which cells harness the reduction of oxygen to generate ATP, the cell's energy currency. Additionally, mtDNA codes for 24 RNAs that are involved in mitochondrial protein synthesis. Most of the proteins in a mitochondria, however, are coded for by nuclear genes. As such, mutations in both nuclear and mitochondrial DNA can disrupt the function of mitochondria, which can have a huge impact on tissues, organs, and organisms as a whole due to the key role mitochondria play in energy production. Because of the importance of mitochondria, pathogenic variants in mitochondrial DNA often affect many different tissues. Most conditions associated with such variants involve the central nervous system and the musculoskeletal system. Mitochondrial DNA disorders are relatively common, affecting approximately 1 in 5,000 people. I was actually quite shocked to hear how common they are. These disorders can present themselves right at birth or later in life due to either a delay of the phenotype or an accumulation of somatic mutations throughout the aging process. If all mitochondrial DNA are the same in an individual, they are considered to be homoplasmic. If there is more than one type of mitochondrial DNA in an individual, that individual is said to be heteroplasmic for their mitochondrial DNA. Most pathogenic variants of mitochondrial DNA are present in the state of heteroplasmy rather than homoplasmy, because many studies have shown that homoplasmy for a pathogenic variant usually results in that cell's destruction. Note that an individual who is heteroplasmic can have a varying amount of pathogenic DNA, so the term does not quantify how much of their mitochondrial DNA is mutagenic. Since most people who have a mitochondrial disorder are heteroplasmic, a reasonable question to ask would be, what proportion of variant mitochondrial DNA is required to result in a disease, or more specifically, a phenotype? There is indeed a threshold for phenotypic expression of disease that is usually around 60%, but this threshold value varies by the variant and the phenotype involved. This means that a person could be heteroplasmic for a particular variant at a rate of 40%, but they do not display the disease phenotype associated with the variant. It is possible, however, that they will have a biological child who inherits the variant in a proportion that does result in a disease phenotype. Generally, the level of heteroplasmy correlates with the severity of the phenotype, the number of tissues involved, and often the likelihood of passing the condition on to an offspring. As a general rule, sperm are thought to not pass on mitochondrial DNA, so a zygote can only inherit mitochondrial DNA from the egg. As such, an individual with a mitochondrial DNA variant will not be able to pass on the variant if they are providing the sperm, but they may pass it on if they're providing the egg. Of course, we can never say never in medicine, as there has been one report of sperm inheritance of mitochondrial myopathy, but so far, this is the exception and not the rule. So, why did I say that an individual with a mitochondrial disorder may pass on the variant to their offspring via the egg? Well, there is a phenomenon with the mitochondrial inheritance called the mitochondrial genetic bottleneck that occurs in oogenesis. Developing oocytes contain a fraction of all the mitochondrial DNA that exists in the body. So in a person who is heteroplasmic for a particular variant, They may have some oocytes that are homoplasmic for normal or wild-type mitochondrial DNA, and oocytes that contain varying amounts of the variant DNA. Furthermore, the actual replication of mitochondrial DNA is independent of the cell cycle and completely random. Therefore, in a given cell, it is possible that more wild-type mitochondrial DNA molecules are randomly selected for than variant DNA molecules, or vice versa. This will ultimately change the level of heteroplasmy in the offspring. Finally, during actual cell division, mitochondria segregate randomly, meaning that the distribution of variant mitochondrial DNA can once again be disrupted, and some daughter cells can even be homoplasmic. For all these reasons, inheritance of mitochondrial DNA-associated conditions is not straightforward, and we rely on genetic counselors and medical geneticists to help families understand their overall risk of transmission to offspring if this is something that they're concerned about. Just like nuclear DNA, pathogenic variants of mitochondrial DNA can either be inherited through the germline or acquired through somatic mutations. As we age, somatic mutations of mitochondrial DNA increase in frequency, and we are learning more and more about how mitochondrial variants contribute to the aging phenotypes. Mitochondrial inheritance is a bit difficult to wrap our heads around, but it is important to be aware of how relatively common these conditions are in the general population. Mitochondrial DNA variants and their impacts on human health exist on a spectrum. For example, there's a common polymorphism that has a small effect on the risk of developing Parkinson's disease, and there's also a very rare but highly penetrant mutations of mitochondrial DNA that are the primary causes of inherited multisystem disorders. Our understanding of mitochondrial disease and inheritance is evolving. So, for now, just make sure you have a general understanding of how its inheritance is different from Mendelian inheritance, and how nuanced discussions about inheritance risk can be.